Hello, and welcome to Meet the Authors. Today we're hosting our third session of this series, an opportunity to hear from and engage with the authors of publications that were enabled by the Open Science Data Repository. Our format today will involve a brief overview of the publication by each author, followed by a couple questions from me, and then we'll open up the floor for the authors to converse either with each other or to respond to questions from the audience. So I'll be supported by the ScienceCom team, who will be putting links into the chat as we go or as needed, and who will also help answer and jury the questions. So today, our Meet the Authors features Dr. John Z. Kiss of the Florida Institute of Technology, Dr. Richard Barker of Purdue University, and Dr. Borja Barbero of Texas A&M University. So Dr. Kiss, if you could please start us off by letting us know a little bit about your publication, The Efficacy of the Random Positioning Machine as the Terrestrial Analog to Microgravity in Studies of Seedling Phototropism. We'd appreciate that.
Yeah, well, thank you very much, and it's a pleasure to be here, and I appreciate the invitation. So the major issue with that paper is we're looking at microgravity analogs, and this is a very important area, I think, in space biology. Spaceflight is very limited opportunities, as we know. So we're always trying to test the efficacy of analogs. And there are different kinds of analogs, clinostats. By the way, I really prefer the term analog. A lot of people call them microgravity simulators. And I never liked that term because you really can't simulate microgravity. And we used a device called the spinner, and it's a 3D clinostat. And we compared it. What was really cool about our studies, we compared it to true microgravity. And I think the more we do things like that, the better off we are. And eventually, we'll see what's the best kind of analog. And we found that for certain parameters, it worked really well, and for other parameters, it worked less well, which is fairly typical. So that's sort of a high level overview.
Thank you, Dr. Kiss. Dr. Barker, could you please give us brief highlights of the publication, Transcriptional Response of Arabidopsis thaliana's Root Tip to Spaceflight?
Oh, the second one. Okay, sure. So the reanalysis of the Kara root tip study was conducted by my friend Mohammed. We were looking at the original data from Rob Foe and Annalisa Paul, and we were looking at the paper, and there's a lot of data there. And in their analysis and in the paper, they didn't really dive down too deep into the response of the hypoxic cluster. And we know that there is a hypoxic response cluster in the root tip, and that this is likely to be responding differently in spaceflight. And so we were kind of like surprised. And so essentially we wanted to revisit that study and then test some different statistical models to see what alternative insights and narratives could be found with different packages. And yeah, essentially, so we did a multifactorial model where we merged together different genotypes and were able to create a different way of interpreting the Arabidopsis response to spaceflight. We then used some new tools, some new code in KEGG to examine the major pathways that were involved, the ones that are most significant, which included circadian rhythm response pathways and the sugar and starch metabolism pathway. And so essentially we were able to revisit this data and also revisit a couple of the genes that were mentioned in the primary paper. There was this one gene called SWEET, this from the SWEET family. And when the first paper came out, that gene was only really known to be involved in transporting of sugar. Since that paper came out, there was more literature that showed that gene is also involved in the movement of hormones, such as gibberellin at this early stage in the root tip, in the developing radicle. And so it was interesting to then see our alternative statistical models still finding that same gene and then being able to provide a different interpretation of what it could be doing because of the advancement in the literature in a different area of research elsewhere on the planet. So it just kind of highlights the opportunity that exists revisiting some of these the great studies that have been performed by some of the researchers that deposit data in the Open Science Archive to kind of show that we can find new narratives within there, new insights that can be used for like breeding efforts and engineering efforts for the future to try and make plants more resilient to the stresses associated with microgravity and the spaceflight environment. So yeah, I think that's a rough summary of that particular paper. There's different tangents within it, but I think that's reasonable.
Thank you, Dr. Barker. Dr. Barbero, could you please go next and describe your publication, Arabidopsis telomerase takes off by uncoupling enzyme activity from telomere length maintenance in space?
Yes, absolutely. And Jennifer, thank you very much for the invite, and it's great to see everyone in here. So I think the motivation for our work that was mostly undertaken at Dr. Dorothy Sippen's lab, as well with combination with Dr. Sarah White's lab, as well as Imara Pereira, we were testing to see what is the effect of a spaceflight on Arabidopsis thaliana telomere length. And this is coming from the fact that humans, when they are on the International Space Station, their telomeres lengthen, which is a very shocking result for multiple reasons. Actually, that telomere length was shortened upon return back to Earth of those astronauts. But the mechanism of why that happened is still unclear, why those astronauts experience telomere lengthening happening. As well, beyond astronauts, there has been another experiment looking at telomere length in other organisms, in this case in C. elegans, back in the 70s, in which as well C. elegans displayed telomere length increase the moment that the organisms got to space. So the question that we had is this something conserved? Is this something that plants will as well experience? If we send Arabidopsis to space, are they going to suffer increased telomere length? And the answer is no. Plants did not change their telomere length when they were in space. And to our surprise, and very interestingly, we saw that, however, there was a dramatic increase in telomerase activity. Telomerase is the enzyme that is in charge of adding telomeres at the end of the chromosomes. So to our surprise, we saw that there was a 150-fold activity increase in telomerase activity roots of the Arabidopsis thaliana samples that we sent to the International Space Station. So then we started wondering why would plants very heavily regulate telomerase activity if it's not going to lead to telomere length changes. And then doing some further ground experiments at Dr. Sippen's lab, we observed that that increase in telomerase activity can protect plants from genome oxidation damage. So now this led to uncovering a role of telomerase beyond telomere length maintenance in space and in protection of plants from genome oxidation. Then we pose another question. It's like, okay, there's a lot of factors, as John Keys mentioned. Microgravity is one of the major differences between Earth and space, and we tested if these changes were due to just microgravity and microgravity alone. So doing some RPM experiments, we observed that that telomerase activity did not change under RPM conditions, under microgravity, when plants were grown on the ground just under an RPM. So obviously our next hypothesis could be that it could be radiation or a combination of radiation with microgravity. So that is something that we're currently following up on, and we're very excited to see results of that coming up soon.
Great, thanks. And thanks for the sneak peek at the upcoming research too.
Yep.
Dr. Barker, could we hear from you a second time on another publication that you were working on, meta-analysis of the space flight and microgravity response of the Arabidopsis plant transcriptome?
Yeah, it's an honor to have a chance to revisit this meta-analysis that I performed with the plant analysis working group. It was a team of us. So I've got to give kudos to Colin Cruz and Christina Johnson and all the other team members who really made it possible. The goal here was essentially to see, could we put together RNA-seq data and microarray data, different types of transcriptional data, and merge them into one overall matrix, and then see what the consequences of using these two data types together is. Essentially, we knew there are issues associated with batch effect in most experiments. It's quite common. And essentially, we wanted to see whether if we use a common computational pipeline, whether we could try and get some of these studies to agree with each other more completely. Ultimately, what we learned was doesn't really work that way. Each of these early studies were designed so exquisitely to like really focus on different factors using different tools and different ecotypes and genotypes. It was really hard to find like a core overlap when you take together all the different types of hardware and like stress simulations that are incorporated into this. So like gamma radiation and random positioning machines were also part of this matrix. So essentially what we were able to do was identify like the structure within the data, which allowed us to essentially focus in on specific hardware types and then look for conserved response in specific hardware types. In that particular paper, we drill down into the brick hardware because it's the most commonly used type of spaceflight hardware for production of transcriptional data on plant response to spaceflight. The veggie system is coming up close in the rear, but the brick is the pioneer, as it were. And what we were able to find when we did that was actually find a core conserved response to spaceflight as perceived in that hardware. And that essentially involved both the cold shock and heat shock response pathways. So there are these core genes, this core family, which are expressed around the rapidly elongating ground tissues, that's like the cortical cells and the endodermal cells. And there's actually, that's one of the areas where we have the most plasmodesmatas in the root tip. And so we think that maybe these genes are involved in the regulation of either opening or closing or plasmodesmatas in that region, which will alter like the hydraulic environment and essentially cause the roots to either swell or not, like elongate or stay short, in fact. So the more the studies we found, we were able to find some genes that are causative of some of the phenotypes that have been observed with this particular way of drilling down into the data. In addition to that, we essentially found that a protein called heat shock protein 101, so it's like a molecular chaperone that can help denatured proteins continue to function before they're essentially degraded in the ERAD pathway, the endoplasmic reticulum associated degradation pathway, which is essentially then investigated in the Open Science Data Archive study like 321, where they essentially looked at mutants that had altered vesicle trafficking and essentially showed that they had a different response to the spaceflight environment, kind of highlighting that in the brick, in this truly hypoxic hardware, there is like protein denaturation that is occurring, possibly because of hypoxia, and those proteins are being processed in the ER. And HSP101 is one of those key proteins that's involved in that. So essentially, this meta-analysis was able to find fingerprints of hypoxia, changes to the oxidative phosphorylation pathway in the mitochondria, which essentially can lead to some of these phenotypes when the mitochondria fail and they break down, they release reactive oxygen species and cause that protein denaturation, which was described earlier. What essentially we learned was the meta-analysis of multiple studies was allowed to provide a more holistic picture of what's going on and reaffirmed our fascination with this cell wall and how that is being altered in spaceflight, because we were describing there the intracellular ROS signaling systems that are responding to things like radiation and hypoxia. But but ultimately, the byproducts of those reactions end up in the apoplast, out in the cell walls, and alter the cell wall structure. And so there's a lot to learn about how the cell walls are adapting to spaceflight and how that is likely to be different in different tissues, be it the cells in the center of the root versus the outside. So so the more of the story, spaceflight can be stressful, but plants can adapt to it. And they do that by changing their oxidative stress response system, altering photosynthesis, altering ion transport, and essentially doing a lot of the adaptive traits that we can observe here on Earth. So there's a lot of opportunities for follow-up studies on Earth to help us better understand these stress adaptive traits, to help us inform future experiments in LEO and beyond. So kind of, reiterating the how this meta-analysis identified opportunities for more ground-controlled studies to help provide more conclusive insights from the flight studies. Because we do need to detangle the hardware-specific effect that can be observed. Okie dokie. Well, that's the main narrative, a reasonable summary of that paper, and feel free to dive in and reach out, and we can have a conversation about it at any time.
That's a perfect segue, because I actually wanted to ask a question about that ground control, but I actually wanted to direct this question to Dr. Kiss. So in light of that, Richard's mentioning the need for ground-based studies. Could you actually please tell us a little more about the value or really necessity of analog experimentation on the different plant tissues, please?
Well, That's a good question. So, I mean, I think the analog studies are are important in two ways. One is they could actually help you define a space flight experiment or parameters you're thinking of looking at. They could give some clues. So that's sort of before, but that isn't what we did, but other people have done that. They're also useful for after because you're going to find some interesting things from transcriptomics, other kinds of data. And ideally, you might want to design another experiment to do it for in flight. And that would be the best route, but you may not have that opportunity. So then you could use those analog studies to try to tease out information you learned from a flight study. But ultimately, I think it does need to be verified by flight. Um, can I make a comment on Richard Barker's?
Absolutely.
Yeah. So I'm really happy they did that meta-analysis because right now there's just tons of transcriptomics and they're deposited. So it was, I think he did a very valuable service, he and his colleagues. Um, but I guess so to be clear, Richard, so do you feel that um, despite there are all these different hardwares that are designed for specific things, a lot of it was brick, and brick really is not a great way to grow plants, right? Because it's um, so was the idea that if, despite all these different studies, if some common uh, genes, pathways emerge, that it's likely to be true. So, for instance, you cited the cell wall. Is is that kind of where you're going?
Well, yeah, one of the genes that's like nearly uniformly differentially expressed across most transcriptomes is the cell wall synthase. So it's kind of that final
Right.
endpoint of like the carbon metabolism. And so essentially, when you're like happy and you're not stressed out, like your cell synthase is out there kind of at Vmax, making your cell walls as much as possible. But when you get a bit more stressed out, that gene expression appears to be going down. So they're not producing as much of those like hydrocarbons. So essentially, the oxidative phosphorylation system in mitochondria, if it's not running at full potential, like this amount of sugars that go into the cell walls will be reduced. So yeah, it's going to come back to atmospheric potential really, because the brick is double sealed, right? So when we're looking at all the different experiments in there, it's there's a finite amount of oxygen that's in there, and that's actually determined by how much sugar you put in. So by adding in more sugar, you can have more oxygen in that system and get a different response. But then when you take the brick away and then you expose to the International Space Station and you have the atmospheric composition there, again, you get a different like primary carbon metabolism response. And so once you can take that into consideration, you can then start to find some core responses that are true in both the brick and in the space station itself. But you kind of have to take into account like the atmospheric conditions as well as like the tissues and genotypes. So we're just getting to the stage now where there's enough studies that similar enough where we can actually start to deconvolute some of these complicated factors and then really start to paint a clear picture of how the different organs are responding in the different hardware types, aka a true hypoxic brick versus with light versus dark versus any kind of agricultural hardware on the ISS which is exposed to the astronaut's exhaust. Couple of different like key distinctions that exist within the data that are sometimes hard to tease out of the metadata because there is so much of it thanks to the work of NASA and then the Open Science Archive and making it available. Ding.
Thank you.
Awesome. Thanks, Richard. On that note, I actually wanted to piggyback off of that and ask Dr. Barker about, do you notice the difference between the types of analysis that are being happening? So, for example, could you just comment a little bit on the difference between microarray and RNA-seq datasets for your transcriptomics analyses?
Well, yeah, I missed that point in my rambling introduction to it. Well, the key difference that we learned was they were both capturing like most of the genetic response to flight. But when we broke down the genes that were differentially expressed and did a weighted genome correlation network analysis. So essentially, it's an AI statistical tool that groups or clusters the genes based on their known biological function, and we can then see, like in the RNA-seq, 5 main like functional groups responding to flight, whereas in the microarray study, we could only see 4 clear functional clusters. So essentially, the RNA-seq was able to capture an extra functional cluster of genes that were responding that isn't visible if you use the microarrays in the model. And that is essentially associated with alternative splicing and all those exquisite alternative forms of RNA self-regulation, like post-transcriptional regulation, because the original microarrays were designed to monitor the primary proteins that are known to be really important. So you can learn so much from the microarrays. You really can. They catch like there is some of the really interesting protein response. But yeah, the RNA-seq just has this extra cluster of alternative like not changes in non-coding RNAs, microRNAs, all those things that essentially cluster together to be like the non-protein coding cluster. And so that was just interesting. We didn't know what we would see if we had these 2 technologies used in the same time or separately. And that was the thing that we saw was what was different when we merged them together. So similarly, when we start to add in like proteomic data, again, you end up drilling down to, again, a smaller part of the response. Again, it gives you more confidence that those nodes are really like functionally important. But again, it really does then focus down on like a smaller subset of the overall response. So yeah, the moral of the story there's a lot of opportunities for multi-omic analysis. There's new studies coming through that have that capacity. But at the same time, you get a nice clear picture from the RNA-seq because it does capture the full transcriptome's response. And I like that because it gives you more like data points to put into your like multifactorial analysis. So statistically, it's strong. Okay, over. Thank you.
Great. No, thank you. Thank you very much. I want to step back a little now, and I want to ask a different type of question to Dr. Barbero. So, as you mentioned in the paper and here in this discussion, you mentioned that there was not much change in the telomere length despite the changes in the telomerase activity. Did you find this I'm not sure if your study went to this, but did you find this in more than one ecotype of the Arabidopsis plants, or do you have intention to pursue deeper dives into this? Because you were talking about, like evolutionary nature and conservation. I'm curious, is this happening in different even ecotypes of the Arabidopsis, or if you'll intend to pursue that?
Yeah, absolutely. So great question. We did these studies in Colombia background, which is probably the most common Arabidopsis ecotype studied out there. But we just sent another payload to space, another launch in SpaceX 32 resupply mission. And then on this payload, we are sending actually different genotypes to it. So our hypothesis, which is to see this increase in telomerase activity could be due to increased ROS. We're sending plants with deficient ROS mechanisms to see if that even exacerbates telomerase activity even higher. We didn't send any other ecotype with it, but because all of it was done in Colombia background, but we're targeting that specific question as well. See if this is something that is maintained within our hypothesis. As well, we're sending plants that have an increased levels of telomerase activity that under as very strong promoter, and see if that increase in telomerase activity will make these plants on the International Space Station be actually lower levels of ROS, just because they have this enhanced capability of perhaps dealing with ROS. And similarly, to test these levels of, to see if telomerase activity is very important to deal with oxidative stress in the space, we're sending Catalyst 2 overexpression plants to International Space Station and see if we see a similar response between Catalyst 2 overexpression and telomerase overexpression in dealing with genome oxidation. So, but that is a great question.
That sounds pretty exciting. You got a lot of stuff going on. And then I just have another follow-up, excuse me, another follow-up on different taxonomies of plants. So I know we're mostly talking about Arabidopsis and pretty much all your papers are about Arabidopsis. But do you think these activities would be different for plants that have different life cycles? Like, for example, if they were non-flowering?
So in terms of telomerase activity, telomerase is mostly expressed in in dividing cells. So this will be mostly at the stems. So for that particular case, I imagine plants that are still not flowering, that telomerase activity will be very concentrated to actively dividing cells. Whether we will see a similar response in other crops, I actually do believe so. Telomerase activity and telomerase is actually very well maintained and conserved across all of the different plants. Whether this activity of telomerase is maintained or is like different mutations and differences in telomerase that make this activity happen in terms of dealing with oxidative stress is to be tested. But I do believe that we will find a similar response in other crops or plants flown above the International Space Station in regards with the telomerase activity.
That's awesome. That's helps my curiosity. But you did mention the plant parts now and the stems, and we've been talking about roots. Could I come back to Dr. Kiss's paper where I believe you found differences between the plant parts, between the stems and the roots, I believe? Could you speak a little bit more on that?
Yeah, I mean, it was kind of a surprising result. So the microgravity analog system we used, the spinner, it really worked well for the shoot phototropism. So the results there, the analog results were very close to the true microgravity in spaceflight. But the root phototropism, which I expected the opposite. That was not. It didn't really replicate or the spaceflight results very well. And I think that's just the general. And I don't really have a great explanation as to why, except for the general point that when you look at these microgravity analogs, and I'll say simulators in quotation marks, it really depends on the parameter, and that's what that you're studying, and that's what other scientists have found with all sorts of microgravity analogs. Is it works great for some things and doesn't work well for other things. It seems to be specific to the experiment. As you pointed out, this is the same plant. It's root and shoot, and maybe the plant species. So I think, I mean, one thing I generally object to is I get these papers to review and they talk about, this microgravity study, and then I look at it and, oh, it's really a microgravity simulator. So I get very annoyed by that. You really need to identify what you're doing. But again, I think it's imperfect, but I think as we develop these, it's good that we have something. And I think, I don't know, but some like random positioning machines, I think there were different refinements on these in terms of the software. By the way, some of them are also doing a topic that's very interesting, is not just trying to do an analog for microgravity, but doing the fractional reduced gravity of the Moon Mars level. Particularly, I've seen that with some of the RPMs. And my good friend and colleague, Dr. Barker, could, I think he and I have talked about that as well. And he's been involved with some of these development of some of these studies.
Yeah, on that note, I'd like to ask all three of you to speak on this. Maybe we can start with Dr. Barker, and then Dr. Barbero, and then Dr. Kiss. But this question here, what trends do you think your papers could illuminate in upcoming spaceflight studies for plants, especially in terms of flight hardware, assay techniques, or other factors? So yeah, if we could get Dr. Barker to start us off.
Interesting trend I think we're starting to see is actually one I think started by Edison Ragafort and John Kiss and their teams, as it were. But the analysis of the cell walls. So in the early days, we'd like get the sample back, we'd grind them up, we'd extract the RNA, and we'd look at what was changed, right? And then a bit of time passed, and some clever people like aforementioned, I realized that we could actually go back to the membranes that we were using to extract the RNA, so that we could and then essentially revisit like what was left over, and that turned out to be the cell walls. And then we were able to extract essentially a new type of molecules. So we look at like the sugar, the glycomics profile that's within there. And that's really, really important because we mentioned, like, as we look across all the different types of spaceflight hardware, the key thing is ultimately there is a change to the cell walls, and it's different depending on the hardware type. And so it'll be really interesting to see what new insights come from upcoming glycomics data. I think Edison has presented the first from that, and hopefully there's a series that follows the other similar types of data. But yes, that's one that I'm quite excited by because I'm fascinated by the cell walls and how they adapt. Okay, that's my perspective. Next one.
So I guess picking up on Richard, I think the last decades we have been learning quite a bit about how plants behave in low Earth orbit. And now we're going to have a more pressing question, which is studying these plants beyond low Earth orbit, where the levels of radiation are going to be higher. And probably, obviously, Richard and John can attest for this more than me. But there has been differences between experiments and between different hardwares and between different rollouts, in which a lot of them the full analysis of the metadata wasn't available for X reasons, and now we know the importance of trying to keep the conditions or understand the conditions for all of these experiments, because many things can change between one flight to the other. In terms of our research with telomere biology, what we were able to see is that the plants, unlike other organisms, they are able to maintain telomere length on that specific range, which is like a healthy range, which promises that plants might be able to endure the dangers that space can entail on organisms. But now, trying to colonize Moon, Mars, and go beyond low Earth orbit, we'll have to ask the same question again: if plants will be able to sustain their genome, maintain their telomere length upon these new flights and missions that are coming towards us. So,
Yeah. So, Dr. Barbero, made an interesting point about teasing out all these very different effects. And, I think, one of the things, there are what I would call the true microgravity effects. Of course, that also, the other topic we haven't really touched on is radiation, because the radiation environment on Moon, Mars, and even getting there is going to be kind of severe. So how does the microgravity environment interact with the radiation? And of course, that has all the radiation that's how we mutate plants. I think that's a very big open question. I think another one, and of course, I read Richard's paper. I guess I should call him Dr. Barker's to be egalitarian here. So Dr. Barker's paper, the meta-analysis is, we found in some other of our transcriptomic studies our effect on light pathways, photosynthesis pathways, chlorophyll, the light harvesting complexes, all these things, photosystems, and there seems to be down regulation, and how does that affect the plant? Is the photosynthetic ability going to be ultimately affected? Could you almost turn it back to Dr. Barker? I just don't recall from that paper what they found. I agree with them on the cell walls. There's been lots of evidence on these effects on cell walls. Of course, the telomere story is very fascinating, but I don't know about the light pathways, which I think could have a super important effect.
I'd like to, oh, you're muted, Dr. Barker, but I want to ask a naive question to tag on to this. So you bring up the radiation. Do cell walls confer different protections against radiation than cell membranes?
Not I mean, I guess, but no, I mean, it's pretty minimal, and it gets through the cell walls, and it could cause all sorts of harmful mutations.
And then Dr. Barker, did you want to address Dr. Kiss's question? I jumped in because I was so curious about this.
Essentially, just the confounding feature about trying to interpret the changes to photosynthesis, again, because of the skew in the amount of studies that are just done in darkness and don't have a light versus dark zone at the same time. So initially, when we were trying to compare light versus dark, when the Matrix paper was first conceived and created, there wasn't really a study where that was the factor that was controlled. So there was always a big batch effect, other than the Kara root tip study. And that's why we ended up doing that deep dive on that particular data set, because it had the light and dark factor. So we could start to try and unravel some of the conundrums that people had associated with the mitochondrial chloroplast function, because essentially that is so in tuned to light, because light being like the primary driver of the response to flight. There's a paper that came out later from Rob and Anna looking at the shoots in the Kara study, where we had this really controlled. But we have one extra interesting confounding feature when we look at that study, and that is in the International Space Station, so it's exposed to the astronauts' exhaust. So that one has relatively high CO2, although that exact measurement, I'm not sure if it's exactly where the sensor is, and that is actually really, really important. And essentially, the exciting thing now is that now we can revisit the brick hardware that we're focusing on too historically, and now there's a new study where we have the brick LED. So again, we can assess that on the samples in there with light versus dark. So, and because that's double sealed, it doesn't have that elevated CO2 background. So there's an opportunity for us to really describe the difference light-dark response in roots and shoots separately with ISS atmosphere or sealed container atmosphere. And I'm leaning into the atmosphere component because of some recent lessons learned. So a lot of us have been inspired by the Apollo astronauts, the Apollo era, and hopefully many more of us have been inspired by the new Artemis program. One of the things I didn't know is when the astronauts were doing the extravehicular activities, their spacesuits are at half atmosphere pressure. So we've got like 1 bar pressure around us now. But in order for the astronauts to be able to move in those spacesuits on the lunar surface, it has to be at a lower pressure. So their cabin was at a lower pressure. And in order to continue to function, and they needed to have the oxygen percentage increased so that the partial pressure of oxygen wasn't reduced as much. So when people are planning the return to the lunar surface as part of Artemis, they're planning to use that same platform, essentially using half atmospheric pressure for future exploration missions. So this has got me like really interested in the effects on partial pressure of oxygen and CO2 and how that would affect the primary carbon metabolism. And so, yeah, so the more of this story is we need more metadata. We need sensors to be put closer to the plants when they're being grown in all different spaceflight hardware. So if you guys are proposing future studies, define where you want your sensor to be. The engineer will only do what's written on the engineer's like tick off list. So if you do not specify that you need to be delivered, say CO2 or O2 measurements from a specific location, that won't happen. You'll be able to get it from some other sensor somewhere on the station, but then you always have that concern about the distance. Similarly for radiation. If you do not specify that you have to have a radiation sensor that goes along with your mission, that will not happen. You will have the chance to try and correlate some of your data with certain radiation sensors that are on the ISS, but there'll always be that little bit of distance and that little bit of stochastic fear, right? So again, when planning future missions, specify these details to your implementation partners. And that's going to be the key difference between you being a bit confused like me now, thinking, hey, maybe it's these other factors, and the difference if you specify that you need these measurements from these specific locations, you won't have that confusion. You will have those measurements to build into your statistical model, and you'll be able to correlate that with whatever omics analysis or high throughput analysis that you decide to do. So hopefully that's some useful advice for the future too, like lessons learned.
Thanks for the pro tips for designing experiments. Now, okay, so on that note, so you talked about light as a factor. So Um, and I think it was Dr. Barbero's paper. No, actually, it was Dr. Barker's paper. You mentioned that light supercharges the root tips. Does the spectrum of the light matter, or will that matter? Because now we've mentioned some of the moon and Mars situations. I believe light looks a little different there from my planet, from planet Earth, where we currently are. Will the spectrum matter for supercharging those roots?
I think so, and that's based on actually John Kissler's work in the European Modular Cultivation System, where they looked at the root tip kinetics in a range of different gravity levels. Correct me if I'm wrong, sir. And some of those were actually the lunar gravity level. And so he was able to show that depending on whether you illuminated them with red or blue light, there was a different response, different growth rate, and I think even the orientation it might have been different. And so essentially, yeah, the light environment that we provide to the roots in the lunar gravity level is likely to change the root system architecture. So now we know that we can try and optimize that, but that's a different experiment for another day. John, how far off was I, sir? What did I miss?
Well, at least I know one person reads my papers carefully, so no, that was quite good. But I'm not sure if actually I think what you're asking, Jennifer, is an interesting question. It might be a little bit different because I, the spectrum. I just don't know much about that topic. So Mars is further away and it has the CO2 atmosphere. So the kind of light that you're going to get on Mars is going to be different than what you have on Earth. That's actually another interesting topic. I mean, I think probably we have enough information from all the rovers that I think we do that we could try to simulate that kind of light, and that might, especially with phytochromes, which control a lot of plant development, that might have an effect there. So you're suggesting an exciting new experiment for me, Jennifer.
Great. And then, so for the audience member, you heard it first here. But I have one more question that I want to ask, but I do also want to address audience questions. But we've circled around this topic, maybe orbited, as you will. But we mentioned the increasing number of lunar regolith studies. Could each of you, maybe starting with Dr. Barbero, just start off and just tell maybe about your studies in context of the future of maybe lunar regolith studies? Go ahead, Dr. Barbero.
Yeah, so I think everybody was and is still in awe of the experiment of Annalisa Paul and Robert J. Farrell of growing Arabidopsis thaliana from genuine lunar regolith brought back from all of the Apollo missions. I think beautiful paper, beautiful work. And then everybody after that has been trying to, try to master that same technique so we can become all lunar farmers. What we observe with the lunar regolith work, sadly, is that the telomere length of these plants grown across different generations on the regolith is that the telomeres are getting shortened. So that means that the genome obviously is losing its integrity, and it gives you, so to speak, a number of different generations that it will potentially grow. Obviously, this was a lunar regolith simulant. We didn't use genuine regolith for this work, but it is pointing us into a direction in which plants' genome integrity might be impacted if we grow these plants in regolith. Obviously, this is where, as scientists, we need to propose ideas, propose different modifications in the genome that could allow these plants to grow if this is the way that we want to grow plants on the moon. And then now, actually, probably I want to do a tiny bit of publicity. We do have a NASA plant AWG subgroup that is starting to concentrate on regolith work. Richard Barker has already prepare a lot of data over these past years, and now we're all collaborating and trying to come up with a beautiful story that hopefully will be published soon through NASA Gene Lab.
Yeah, I think that I haven't really done regolith work myself, and I think a colleague here at Florida Tech, Drew Palmer, is doing work. I think he's part of your lunar regolith group. We actually might have a lunar regolith meeting here at our university, but there's some pretty good simulants out there. And there's actually a company here in Central Florida, I'm forgetting the name, but uh, they are making
Exolith.
excellent. Thank you. See, they're making this simulant, and a lot of people want it. So I think that's a good thing. And I think there's going to be more applied research like that, and that's pretty much all I can say. I mean, I've never been brave enough to grow full plants. I mean large plants. So I work with seedlings, and seedlings have a real advantage of it's much easier to grow seedlings than to grow a whole plant. But I think that's sort of I think there are a lot of regolith studies. I'm glad to hear about that regolith group. So I think we're going to have some interesting stuff that's forthcoming.
One little tangent. I saw an awesome presentation just a couple of days ago, or I say, at the plant analysis working group from an amazing high school student from Chicago, Adia, and she actually put a concept forwards in my mind I hadn't considered before. She looked at the different like lunar samples, and she looked at their like chemical composition and their kind of ratios on it, and then she realized she could just get those like chemicals from like the chemistry lab, and then made her own kind of combination of powders. And if you've played with the commercially available regoliths, you often see that they're kind of powdery anyway. And that is probably the biggest challenge about growing stuff in them often, because it's hard to grow stuff in a powder because of the soil structure. We won't get bogged down in that. But ultimately, what I realized, what I really appreciated in her work was because you got the chemical components just right. If you could suspend them in like a media and then do it as like a nutritional like assay. So I'm a bit old fashioned. I'm an Arabidopsis scientist. So I want to grow them in their natural environment, a square petri dish. And here with like essentially with the recipe that she presented, you could then create a custom lunar regolith with a gel format, so to allow us to kind of drill down some of these like mineral narratives. But anyway, so always learning, and that's why I'm really excited to and honored to be part of the Parnassus working group. And every week this one that turns up with a great idea that sparks off new conversations. And
Yeah.
so,
Richard, we actually performed something similar to what you're mentioning, making an agar with regolith composition. Ended up very messy, but it was quite fun to see it. And yes, the presentation from Abilla, I think was her name, a high school student. It was amazing. So
really refreshing. It's cool, so cool.
I think your conversations have supercharged the audience. And so there are several questions that I want to make sure we address. So I just scrolled up into chat. The first one was about, I think this is directed to Dr. Barker. They're curious about how polar charge electron availability affects cell movement and wall, and how is that different in microgravity versus the 1G environment?
Yeah, and that's going to be particularly fascinating in the organelles I alluded to earlier, the chloroplasts and the mitochondria. I mean, there's electron transport that's going through the membranes usually, but in those organelles, it's kind of like hypercharged because they're using it to create proton gradients to create ATP and the likes. So, polar charge electron. So the more of the story is it's really hard to isolate the microgravity component in flight because we're going around the Earth, we're going through the South Atlantic anomaly. And so there is this like weird proton flux. So things as sensitive as that are really hard to kind of describe with the source of data that we have. But on Earth, using like RPMs to create simulated microgravity, there could be ways to investigate that. I actually think it is a really interesting area. And I don't want to get too hypothetical on the sort of tangents that it puts into my mind. So I just say, hey, Carl, we should chat about that sometime. And it wouldn't be really interesting if to put a RPM in an electromagnetic field and see how that system you're describing would change. Kind of, or to be continued. So it wasn't a good answer.
You have another, oh, I'm sorry, go ahead. You have another question or a comment from Terry Trevino. We're keen to study how seed production progresses for some of our tests. Will we see a stressor response with higher seed production? And he knows that he saw good production in microgravity and with algae. And was this one directed to anyone?
Okay, I guess I can try to tackle that one. So I guess it's seed production in space. I mean, I think it will be really dependent on what crop you're discussing, right? It looks like the consensus for most plants aboard the International Space Station, it is increased oxidative stress. So obviously the plant metabolically is going to be using probably a lot of energy to try to decrease those levels of reactive oxygen species. And potentially that energy is not going to be used in creating more seeds. That will be a very general question. Per se, obviously as well, there's like the physiology of the plant. Obviously, some plants are self-pollinating, some are not. So those ones, obviously, with microgravity pollination is trickier as well. I haven't done seed-to-seed experiments, but that will be my answer. It depends energetically. Probably the plant doesn't want to concentrate on that, and then there's the physiology aspect of the pollination for each of these different plants that needs to be taken into account.
Yeah, I'd echo that sentiment and say really interesting to revisit that question with like duckweed. I know the duckweed's been going on orbit a couple of times, but if the algae grows better, maybe the duckweed does too. um,
I mean,
uh,
an algae like has like a very good mechanism for stress response related work. So some of the work by Agata Supanska that she has been able to literally hammer some of these different algae with increased levels of radiation, and they display lower levels of ROS, a point and assess that probably algae and so duckweed that they are resistant crops to some of these environmental stressors, and those ones probably will be like home runs in terms of production of next generation.
Thank you. I have a question that's addressed to Dr. Kiss from Sigrid Range. One of the problems with clinostats is the lack of standardized parameters for different organisms under different conditions. Do you have any recommendations for how to develop those standards?
Not a really good one. I mean, I guess I don't know if one of these working groups or various plant working groups. I guess that would be valuable if they came to some consensus. Of course, they're different. There's the classical 2D clinostat, 3D clinostat random positioning machine, and even like the random positioning machines. I mean, some of those, the softwares are proprietary, but I hear the concern because you're right. There are different ways to run these instruments, and particularly when there's software. Um, and of course, the interesting development there is the software for the reduced or the fractional Moon Mars gravity. But I don't really have a great answer unless we form, one of these working groups tackles that and said this is the recommended parameter. But scientists tend to be very individualistic. They'll think this parameter is the best, so it might be hard to get them to agree. But I could also punt that to Dr. Barker because he's thought a lot about these instruments as well.
yeah, there is a paper. I'm just trying to find a link to it so I can drop it in. That describes what they call the Bond criteria. And so it came out like 8 years ago. But it basically outlines what's the minimal amount of metadata that's required in order to make sure that your study is repeatable. I think it's based on like human cell cultures and RPMs. But revisiting the Bond criteria is a good idea, but it doesn't actually have enough criteria if you're looking at plants because plants are exquisitely sensitive to light. And if you're removing the gravity vector, the next like stimulus is light. And again, so I don't think there's any particular RPM study ever that's like described like the light environment or the vector that it came from in a way which is biologically insightful from the plant's perspective. And I think that's really important. Circadian rhythm is a real thing. Like all things are trained in it. And if we're not reporting like all the light details associated with the environment where the microgravity analog is being performed, then it's hard to be sure whether the changes that we are seeing is because of the time of day relative to the light cycle in the area, or actually truly because of the microgravity simulation. So the more of the story is I concur, John. I think there is a need for a revisit or of a meta-analysis of the stuff that's available or the papers that are coming out there, and with the goal of trying to clarify what we think are what is the Bond criteria. I need to find that link to that paper. I'm sorry, I'm going to search for it and try and drop it in the link. Because yeah, there's because if you do a meta-analysis, so you take together all the different microgravity analog studies, and then you look at all the genes and things that changed in that. You have a larger list than you do if you take all the true microgravity studies together, look at all the genes that changed in that. So there's more variation in what we see in microgravity analogs than there is in flight. Kind of makes sense. Sorry, go for it. So good. How's it going? Your hands up.
Yeah, so you have experience with the facility at Kennedy. What kind of recommendations do they give to the researchers who are using their microgravity analog facilities? Do they give you any counseling? Do they give you, help you come up with recommendations, etc.?
I haven't been the researcher, so I can't really comment on that. But like historically speaking, it is up to like the biologist to define like the parameters or the requirements. And that is actually the hardest part, right? Like it's a different type of like language to describe like the engineering associated with like an RPM, and then it's a different type of language associated with how to use one and conduct experiments within that. And so it's a really difficult thing. It's so interdisciplinary. And so the best thing that we can recommend is that if proposing two said microgravity analog research facilities, to try and be as really clear as possible in your experimental design. A lot of people just want to just throw their samples in a microgravity analog and just see what happens. But you've really got to have a really strong hypothesis focused on like a specific tissue or specific response that you expect. Because if not, when you conduct the experiment, you'll see that you should have controlled for something other, like the light environment or the atmospheric environment. So be more specific with your requests when speaking to implementation partners, because generally speaking, they want to know what the scientists want so that they can create that resource for them. It's just really hard to kind of predict what you want because all of your experimental ideas are so unique and different because they're all like pushing the boundaries of human knowledge. So you can do the whole field a favor by actually pivoting around and focusing in and writing down exactly what your requirements are for things like microgravity analogs, then going to said facilities to make them create the one tailored for your need. So for example, like a human researcher might be really interested in the lung response to microgravity. So they're gonna have to have like some really like custom like lung pumping system internally, which you just wouldn't need if and so the normal RPMs never have that type of stuff. Like similarly, a plant scientist might need to have lights at weird angles because their experiment on the space station changed the orientation of lighting. Hey, John. So the moral of the story is, yeah, the community here has an opportunity to really like specify where the field goes. There's it's still evolving, and I think there is a plant analysis working group, meta-analysis forming in that area. Yeah, I think Jessica Baines and who else was it? And someone else is putting together a and so hopefully that'll be promoted within the AW space because we know it's quite a popular area and it's good to get lots of perspectives on that. Okay, so I was rambling, but I don't know if I answered the question, but hopefully it was fun or not. Or insightful.
I'd like to close out our audience questions with this question from Paula Castaño, because this is really what brings us together. And so she thanks you for your insights. But speaking of advice for the future, she'd love to hear your thoughts on how the availability of all these data sets in OSDR is concretely informing future and more standardized experimental designs. And I feel like this is a valuable question because this is what brings us together, and this is what we're talking about now. So if maybe just one of you could touch upon that or address that, that will be our last audience question for this session.
I've talked enough. Like I've talked too much as well. Okay, go
for it. I think the words of wisdom from John and Richard on this matter will be better, but I can tackle this question. I do think, especially with all the amount of data that is available, the fact that people from all over the world can access it and try to come up with their own conclusions, it is extremely valuable, especially not just for analysis of the data, but for formulation of hypotheses for upcoming experiments. So obviously we live in an era in which spaceflight is becoming more and more frequent. However, it is going to be we're going to need to understand some of these questions a tiny bit quicker if we are to colonize all of these different places and we want plants to be an important part of those missions. So I think it is extremely important that everybody comes together, analyzing data, having that data you generate be open access, and people can as well interpret and even support your hypothesis and your conclusions.
Thank you. And I think that's a beautiful closing thought. And so I'd like to thank the audience for being here, thank our panelists once again for the time you've given. We've mentioned a few things, including the plant AWGs. So I'm going to share my screen to let you all get that copy of the QR code if you want to continue some of these conversations with these authors and beyond. So thank you again very much, and hope to see you at the future Meet the Authors.